Okay, so today it's really a great pleasure to introduce Tatyana Sharpee. She's visiting us from the Salk Institute. So Tanya got her master's from Ukraine National University and a PhD from Michigan State, both in physics. I actually first got to know her work through her work in retinal coding, and there she did really elegant work looking at populations of cells that basically respond to the same stimuli, but with different thresholds. And she took a really elegant information theory approach to this and asked, if you have a population of cells responding to something, where should you put their thresholds? And what she showed is that there's a transition as you increase noise, where at high noise, they should all have the same threshold and they should all measure the same thing. And you should just average the measurements. But as noise gets smaller, there's a real critical point to separating into to two thresholds where different cells should make different measurements. So that's one example, but if I could say there's a pattern, Tanya is very good at finding a really interesting problem in neuroscience and biology that has really deep mathematics, not just attached to it, but really underlying what's interesting about the problem. That's really a rare skill. And I think we'll see that here. So with that, let's all give Tanya a warm welcome.
Thank you, Ben, for the kind introduction and for the opportunity to present our work here. I look forward to the discussion, and it's a pleasure to be in the physics audience, and where I hope many people share my enthusiasm for hyperbolic geometry. And I will share with you why it is highly relevant for biological systems. So before going into the details, I want to my slides, please. I need slides. Can somebody help me find my? I can rotate the screen, but the slides are changing here.
Is it possible the mouse is at a different screen?
Yeah, let's do. I need slide. I mean, okay, how about that? Okay, all right. Okay. So I would like to share with you 3 reasons for why hyperbolic geometry is important for biology. And I will present data that actually is a growing thing. It's not a static thing. It's a hyperbolic geometry will grow with information. But as a way of introduction, 3 reasons. One is a very practical reason. So, but they're related. The practical reason is that many data sets in biology have a hierarchical structure. Genes are not created equal. One gene turns on many genes. So the one that is kind of a controlling gene, a transcription factor, or something, a regulating gene that will control many other genes, they're not equal. So every time you have a tree-like structure in the data, it's not just in biology. There is a lot of interest in machine learning now, also for the same reasons, because you have this like a tree-like structure in the data. It might not be apparent, but it can be hidden. So every time you have a tree-like structure in the data, we can start thinking about, if I want to create a map of this data, then the hyperbolic geometry provides a continuous approximation to the tree-like structure. So for example, if, for example, this is a phylogenetic tree, this is some species that gave rise to other species, and I want to evaluate distances between them, I will go to the last common ancestor and back out. So in approximate terms, in technical terms, that can be a Poincare half-space model, and the shortest trajectory, the geodesic, will be attracted to towards the root of the node. So that's a practical reason. If the data is hierarchical and we are trying to find continuous approximation to it, find a map of some kind, then maybe we should use hyperbolic metric. We can still optimize over the curvature and dimensionality. That's a practical reason. Well, this is another way of representing the same hyperbolic space, but also in biology, if you go to biological talks, then they will represent data as clades. How many people know what a clade is? No? Okay, so that's a biologist's view of Poincare half circle. So the clades, there will be some proteins and they will be like shooting out like this. So literally this spherical representation. So when I give talks in biology seminars, I'll say, well, see, this looks like a clade. So, but maybe here, yeah, you can say it's a Poincare circle. So with a density of points uniform in the plane, but actually the infinity is unattainable. So the infinite plane is squashed into the circle of radius 1. And these are some examples of geodesics. And again, the distance is a geodesic that is attracted to the center. So that's a practical reason. Because many data sets are hierarchical, when I want to create a map of this data, one can use hyperbolic metric or think about it. Then there is a more biological and more fundamental reason. And I can recommend a series of papers by Dmitry Kryukov here and collaborators. And they are pointing out that networks that have a hidden hyperbolic geometry, they allow for efficient routing of information. So for example, in this paper, very interesting paper, Sustaining the Internet with Hyperbolic Mapping, they say that already, if you want to communicate in a large network, technically, I should know complete connectivity. Each node should know the connectivity path of the whole network in order to find the best path towards the destination. But it is not practical because nodes go up and down, and especially in a biological network, say neurons can die, can be added. The connections between them can decrease or increase. So instead, what they argue that you can, if the network allows for hyperbolic mapping, then I can navigate using only local coordinates, meaning I know my coordinates in the hyperbolic space. I know coordinates of the target node, and I know coordinates of my neighbors. So I send the signal to the neighbor that is along the geodesic that is closest to the final destination. And using only local information, they can achieve efficient communication in the network. So this is a series of papers, but again, for biological systems, this is essential because again, you cannot know the connectivity, but the connectivity is changing, is subject to change. So I would say that actually these two reasons are related. We could say the reason the data is hierarchically organized is because it needs to achieve efficient communications in the presence of changing connections. And in fact, there's another series of paper by Ilya Nemenman and collaborators where they show that if a network has this hierarchical structure, then you can achieve efficient transfer of information from the outside to the into the network, which is related to the transport problem, where it is information between different parts of the network. So this is two reasons, but they're related. It's possible that biology is organized in a hierarchical way because it needs to achieve efficient communication. But given the fact that it's organized in this hierarchical way, we might as well model it with hyperbolic geometry. There is a third reason that is, I would say, more of a speculation, but I think it's all a consequence of time delays and the finite speed with which signals propagate in the nervous system. For example, the speed of propagation is it takes a fraction of a second, which is substantial. So we see these hyperbolic effects not because of the speed of light, but because on a much smaller scale in the case of the nervous system. So today I will provide you with 3 examples. So for the past few years, we have been using hyperbolic geometry to characterize a variety of biological networks. And I will not go through all of these. I will go through examples 2 to 4. But we found hyperbolic geometry, for example, in olfaction, when it characterizes metabolic state of a plant. So for example, we just had food, and you smell a certain way. So that's an indication of what's happening inside the solid sample. And it turns out that these volatile molecules are subject to a 2-dimensional, kind of a 3-dimensional hyperbolic space. And today I will describe 3 examples. Most of the time I will spend on neural representations, because here we will talk about how this hyperbolic geometry grows proportionally to the information that is received. And then there will be 2 examples, one in gene expression and another one, a short one slide in COVID evolution. So onward with the neural responses. As a way of introduction, there are different parts of the brain that respond to different things. We will be studying data from a part of the brain called hippocampus. It's in the middle of the brain, and their neurons respond to points in the environment. So this is a schematic of a mouse that is running around, and every time a mouse crosses a certain region of space, that neuron as a detector would line up. So a neuron is said to have a place cell. Okay, so that's a statement. And then turns out that these place cells for different neurons come in different sizes. Some are big and some are small. And I will argue that this is actually consistent with hyperbolic geometry. For example, I had to travel from California to here, but I do not maintain a detailed representation of space everywhere. All I need to know is how to get from my house to the airport. Then there is compression of space. And then I need to figure out how to get from the airport to hotel and then here. So, and many transportation networks have this trajectory. So for example, you need to navigate somewhere. You don't go straight where you need to go. You go from the local street to the freeway, then along the freeway, and then from a freeway to a local place. So one could think that this is an example of hyperbolic representation. But it turns out that when people record place cells in this kind of a spatial map in the hippocampus, they were awarded Nobel Prize a few years back for this work. So it turns out some place cells are big and some place cells are small. Now, it turns out there is an embedding that one can do by saying, and apparently this construction was what Charles Darwin used to construct the tree of life. So before, you might, apparently he knew it. So before Darwin, species were characterized as overlapping circles. You know, how much, this has 3 petals and this has 4 petals, but the petal shape is the same. So you could characterize species, and people played games in parlors of who can characterize species better using these overlapping circles. And apparently, Darwin knew of this embedding, that you take the coordinates of the circle as they are, and you take the radius of the circle, how big is it, and you use it as a z coordinate. So you embed from 2 dimensions to 3. And if one circle is completely inside another, you draw an edge. And if they're partially overlapping, it's a dashed line. So basically, with the construction of these place cells, one can create a kind of a hierarchical network. And one of the predictions is that the number of these small place fields should increase exponentially with the size. So we will actually not be using this Darwin's construction, partly because the reality is a little more complicated and each neuron can have multiple regions. So we will be using something else. But this is a mental picture that goes along. So the data that we analyzed comes from Albert Lee, who was at Genelia Farms, Genelia Research Campus, and they were interested in very large spaces. So it's a 50-meter track. I don't know how many meters are from here, but they couldn't fit this 50-meter track into one big room. So the track is kind of crumbled inside the room. And actually, we will use the deviations that were in this track to study some aspects of the data, because the animal runs slower in the more curved tracks, and therefore the hyperbolic space will grow more when it runs slower. But anyway, so you have this large track, and you can unroll it on this x-axis, and each tick mark is the region where that neuron is responding. So you have some cells that have large extent and lots of little ones, small ones. So if you plot the distribution, you can see that it is the tail is exponential. So this is first hint that maybe the data has hyperbolic geometry. But as I mentioned, the reality is a little more complicated because some neuron can have multiple response regions. So this construction is with caveat. So instead, we will use just the correlation coefficient between activities of two neurons. And this will create a distance matrix. So one neuron goes up and down, and another neuron also goes up and down. And you can compute correlation distance between them, and that's a distance. So in a data set of many neurons, you will have a distance matrix. Once you have this distance matrix, you can ask, if I put these points on different surfaces, which surfaces will give me distance with the same statistics? And there are many ways of doing this. You can use multidimensional scaling. You can use topological methods, which we use here, or we have options. So in our case, so the idea is you generate distances between neurons, as in the measurement, and you put points on different surfaces, and you compare two distance matrices. There are multiple ways of doing this. We use the topological method. Has its advantages and disadvantages. But the idea behind this method is that I'm taking a distance matrix, and you can slice it at different levels. And whenever you slice it, you say, if it is stronger correlation than a threshold value, you draw an edge. And if the correlation is weaker, you don't draw an edge. And then for a given criterion for what you consider strong and weak, you will have different connectivity matrices. But at each criterion, you can count how many cycles they are. So for example, if you look here, 1, 3, 5, and 6, they form a cycle because 5 and 1 are not connected. So topologically, it's a whole. And so then they would plot, as you change this threshold, initially only 2 points are connected because this threshold is the same. So the network is not connected. And then finally, as you lower the threshold, more and more, the network becomes more and more connected. More and more cycles appear. And then at some point, everybody is connected, so there are no cycles. So the typical form of this Betty curve that they're called has a shape. It has to reach a peak and it has to come down. So, but exactly where it reaches the peak and exactly how thick this peak is, it all can be used to characterize the underlying structure in the distance matrix. So we use this paper from Vladimir Itskov and Karina Kurta. They showed that using this method, you can discriminate random matrices, which is shown here, from geometric matrices, matrices that are derived by putting points on different geometries. And for example, these are different Betty curves. And in this case, the magnitude goes up with order. So this is circles. These are like pyramids, and these are high order complexes. And in the geometric model, they come down. So they use Euclidean geometry. We use hyperbolic geometry. And one could tell a difference depending on, use it as a signature for what kind of geometry is it. Are there any questions? Yes.
When you're doing the thresholding, are you keeping the longest distances or are you keeping the shortest distances?
We keep the shortest distances.
And that corresponds to larger correlations?
Yes. There are always there are always nuances. For example, what to do with negative correlations. We've taken absolute value. Fortunately, there are not too many of them, but yes.
Are these curves assuming that the points are uniformly distributed in these spaces, or just that you can spaces?
So these curves are assuming uniform distributions, but you can have also non-uniform distributions. And in olfactory work, we did not have uniform. In this case, it was a shell, a hyperbolic space, but all points were around the edge. So in this case, they're filled. It's, I mean, after a while, you start looking, when you look at it, you say, oh, it's a shell. And in this case, oh, this data, this practice, it has different signatures, and it will not come down as in a different way, depending on whether. And then if the data is non-uniformly distributed, then the Betty curve might have multiple peaks. Yes.
This is very recent form. A distance matrix is almost negative definite, right?
Yes.
Whereas the exponential, hyperbolic exponential of it is a positive definite matrix. So is that related? And it seems as if that's a very direct connection between the distance matrix representation and a positive definite.
Yes. So the motivation for the original motivation for using this method is that it was supposed to be invariant to a monotone transformation. So whether it's a distance or e to the minus kind of distance, technically they're supposed to be the same. Because the reason is that because this method is based on rank ordering. Okay? So, and therefore, the original motivation for using the topological method is that imagine that these distances are affected by some nonlinearity in the measurement. And therefore, if I just do a subtraction of these matrices, maybe there will be an artifact. The caveat turns out that, yes, it is monotone invariant, but only if there is no noise. If I add noise, then even though it is monotone invariant, but noise will jiggle the, and we have to add noise because there is noise in neural data. And if we don't add noise, we're never going to match it. So it is monotone invariant with an asterisk that the amount of noise shouldn't be too large. Okay. So now this is comparison with data. So this is these Betty curves for neural data. And what is shown in the dashed line, same in both panels, is the experimental Betty curves. from the data. And a solid line plus the spread is what you would expect if you put the same number of points in different geometry. Here we put them on a three-dimensional hyperbolic geometry. And here we put them in a three-dimensional Euclidean geometry. And one can clearly see that Euclidean geometry doesn't fit the data. But you can fit the data with hyperbolic geometry by adjusting the radius or curvature. And there is some quantification. You can quantify the mismatch between these curves, looking either for L1 distance between the curves or the integral of these curves. So this is a more quantitative evidence that hierarchical hyperbolic structure better describes the neural responses. Turns out that there are multiple data sets, and one can look not only at the linear track where the animal runs around in a line, but also on a smaller scale in a square box. So this is data from Yuri Buzaki's lab, publicly available at CNS, and the same three-dimensional space accounts for neural data. Again, neural data is in the dashed line. The theory plus spread here is here. These are Betti curves of order 1, 2, and 3. And again, Euclidean geometry does not account for the data. Okay. So now back to the growth. So it turns out that so the reason we looked at this square box data is that it had an aspect to it, both of them, but the animal is brought time after time into the same environment. And then it turns out that if you do this analysis, this radius of the space in units of unit curvature is slightly larger for a subsequent introduction to the space. Now I'm told that whenever a theorist is giving a talk, there will be a logarithm. So this is my logarithm here. So, you know, it's like you look at it and it does fit like a logarithm. And what is interesting is that if you look at, if you imagine that you accumulate information in discrete chunks, as in, say, for example, Poisson process, then the maximum amount of information that you can acquire in these chunks of time, delta t, the information will go as logarithm of 1 plus number of observations. So observation t over t naught, where t naught is the measurement time, plus corrections. So in the limit of large observation times, this term becomes, goes to 0. And you're left with log 1 plus t over t naught. So what we are observing, and it's not a fit, but that the logarithmic growth here is what you would expect from a maximum entropy acquisition from the environment. And the new part for, in biology, sometimes people ask, is the environment novel or familiar? So by fitting this logarithmic curve, you can answer that question because you have log of 1 plus t over t naught. So this time t naught can tell you when it is novel and when it is familiar. So for small times t less than t naught, you have a rapid acquisition of information. And then for t greater than t naught, it saturates. So I would say that that's the discriminating when is the learning is new, and then when you are accumulating less information. So that's one statement. And second, it turns out that the size of in the brain, we have finite number of neurons, and one can actually match the number of neurons to the observed curvature in the following sense. Imagine that we put in a simulation different place cells of the kind that match with the statistics match to the experiment. And we count how well does this collection of these response regions can tell where the animal is located. So this can be done with Fisher information. And then we generate these place cells, but suppose you're limited in number of neurons, 50, and you put them into hyperbolic spaces like of different radius, meaning either a network is very refined, has many steps, or the network is more coarse. So it turns out there is a peak that depends on how many neurons are there. If you build a very detailed network, so radius 16, it means roughly I need 2 to the 16 number of neurons, then I build a large network, but the number of neurons is not sufficient to probe it. On the other hand, if there is any hierarchy, then I'm not taking advantage of the potential hierarchical structure in the data. So that leads to an optimal radius for a given number of neurons. And then the more neurons you have, the bigger is this radius. And so for 50,000 neurons, the optimal radius, there is more information per neuron, and the optimal radius changes. And if you plot it as a function of number of neurons simulated, as a function of this optimal hyperbolic radius, you get a straight line here. And an interesting thing is that this is the curvature that was extracted from the data set I showed. So this is the number of neurons in that part of the brain. And that's the curvature that was extracted. So 2 independent measurements suggesting that the curvature of the representations that is observed is optimized for the number of neurons that are present in the brain.
So it's logarithmic.
Say it again?
Logarithmic coordinates.
Logarithmic. And then a little bit coming back to the, so this is a curvature on logarithmic growth on large time scales. But remember, I told you that we are going to use the fact that the track is curved. And it turns out that some tracks are, even though are more curved than others. So when the track is more curved, the animal runs slower. When the animal runs slower, turns out that, so this is the speed. The faster it runs, the coarser is the representation. So this field size on average is larger. And if you extract hyperbolic radius, so this is temporal familiarity, meaning inverse speed. So the more time it spends, the larger is the hyperbolic radius. So of course, this is a little bit the data. We are pushing the data to the limit of what it can sustain. But the indication is that even on the time scale of seconds, the space is changing. So, when you're learning something, they say, take your time. The motivation is that one has to allow time for the biochemical processes that form memories to allow for the growth and the formation of these hyperbolic representations. Any questions? Yes.
So you had capital T over T naught.
Yes.
I presume that the equation that you wrote from Belek is valid for any T over T naught, small or large.
Yes.
Okay, so what versus T naught? What was T naught in this experiment?
I'm not that confident in the measurement of T naught in this graph.
But when you're saying that animal runs slower and that decreases capital T as far as I understand.
Say it again here.
Yeah, if I look at the equations that I think you're using, right? So there's capital T.
Yes.
T naught.
Yes.
Understanding that if animal runs slower, then this ratio becomes small.
Yes. So when the T is smaller, then this observation time is smaller. Yes.
Right. So I'm just asking now, what is the
yes.
Is it 1, 10, or 0.1 or?
So I would say about 1.
1.
Yes, about 1 and less than 1. So it's kind of, it is limits to the data, but whether, so this is fitting log of, you know, kind of in the more of a linear regime. So it's not, there is an additional question that we couldn't quite answer. So there is also, I think I took out the slide, but there are two confounds. One is time, and the other one is how interesting is the track. So it's possible that there is a higher entropy in the environment along the more curvy tracks. And in this particular data, there was no additional effect of how curvy the environment was beyond the effect of time. But in the future, one could say or investigate whether there's a coefficient of log of 1 plus t over t naught, whether it's modulated by the environmental content. Yes.
I have a question about how the hyperbolic radius increases. Is this, should I interpret this as the smallest length scale that they encode gets smaller, or that there's more neurons, or?
So, so I measure the size of the hyperbolic space in units of inverse curvature. So either you can say the curvature increases for a given size, or the radius increases for the fixed unit. But you're asking how mechanistically it happens. So I have actually an answer for that. Oh, I guess the slides did not get taken out. So they're here. My presentation is not synced here. So, so what do you want me to answer? Previous question or this question? All right. So I will ask the previous question and then this one. So this one says the speed depends on the entropy of the track. So the curvier the track, the lower the speed. But it also could be that it runs slower. This one says hyperbolic radius as a function of how curvy the track is. So increased entropy increases. But you could account for increase in hyperbolic radius as a function of time. And different symbols correspond to different curviness of the track. So in this particular data, the slope of this log of 1 plus t over t naught was the same. And the time was the main factor. Yes?
What do you mean by entropy of the track?
Yeah. So the entropy of the track, so this is delta theta, how much it turns, the distribution of that delta theta. Okay. It's a poor man's entropy of the track. So, you know, it's at how much the angle is changing as you're moving along.
Are these
Yeah.
experimental animals with implanted electrodes?
Mm-hmm.
And how many neurons are being monitored?
Depending on the session, about 100.
For the changing hyperbolic radius, we interpret it as a learning. If so, does it sustain around 24 grams, or it's just like adaptation to different regions?
So the question is, how should we interpret the increase in the hyperbolic radius, whether it's learning or adaptation? Is that the question? I don't think we can distinguish learning from adaptation in this context. And actually, the title of the paper says with experience, because we cannot, learning has a specific meaning in neuroscience. So in this case, they were not performing a specific task. So technically called experience. Now, I think that's a good time to transfer to Ben's question about what happens. So what happens is the following. Imagine that with time, the representation gets more detailed. So you either recruit new neurons, most likely you recruit new neurons, but also it is known that the threshold of a given neuron increases. So these place response regions become more selective. And one can actually make a phenomenological model one could make a phenomenological model by saying that we're going to use T0 again. So given the overall observation time T, I should, each neuron should have, should justify its existence if there is a minimal amount of time that the animal spends there. So n times T0. Then n goes as roughly e to the radius. So then T over T0 becomes e to the r. Or in other words, hyperbolic radius goes as log of T over T0. So in other words, if I start adding response regions, the moment can everybody see this? Yeah. Okay. So there's a minimal amount of time that the animal has spent in that region. From this, you will get a logarithmic growth of the curvature with time. Yeah. And I think there was another question. No? Mm-hmm. Okay. Right. Okay. Thank you for your questions. So, and we also seen that the size of the hyperbolic representation is optimized for the number of neurons. So now the shorter parts of the presentation is about gene expression, and then one slide on COVID evolution. So I told you about advantages of topological methods, but they also have disadvantages. One disadvantage we discussed, that they are based on rank ordering, but not quite. And then the second one, there was a question about how many neurons, about 100 neurons. Well, it turns out that you cannot compute these Betti curves for thousands of samples. So 100 cells, this is about the maximum for where we can compute these topological characteristics. So now if you have larger number of samples, as in gene expression, it can have thousands of samples. So now this is a canonical multidimensional scaling. You're given points, you don't know their locations, but you know their distances. You perform multidimensional scaling to find positions, but respecting the distances. And that's a Euclidean MDS, and it works. Now you can do the same thing with hyperbolic MDS. You put, this is in simulations, you put points on the hyperbolic space. You adjust the positions to recover the distances. The algorithm runs slower, but it still gets the same distances. Now you can have, this is multidimensional scaling, but there are two versions. One is metric multidimensional scaling and non-metric multidimensional scaling. So it turns out there are advantages to using non-metric multidimensional scaling for these purposes. So in a metric multidimensional scaling, the goal is to obtain a straight line before embedding and after embedding. In a non-metric multidimensional scaling, the goal is to reproduce the rank ordering. So the curve the algorithm is not penalized for producing a curve that is not straight. And now you can use it as a detector of geometry. So if you take Euclidean data in simulations and you put it in Euclidean space, then using non-metric MDS, you will get a straight line. But if you use Euclidean data and you put it in a hyperbolic space, you will get a line that curves this way. So it has a negative convexity, and the distance between curvature of the space you're putting the data into from minus the initial curvature is negative. If you do, if you take hyperbolic data and you put it in hyperbolic space, you'll get a straight line. If you take hyperbolic data and put it in Euclidean space, the curve will bend the other way. So you can use the mismatch in this in the curve of the fittings as an indicator of the difference in curvature between original space and embedding space. And so by varying the curvature of the space that you're putting the data into and tuning it to get a straight line, you can determine the curvature of the best curvature of the resulting space. So it is a little confusing, but we have to use non-metric multidimensional scaling with hyperbolic metric. Okay, so it turns out that here are some simulations before we do real data. Left is simulations with Euclidean geometry, and right panel is simulations with hyperbolic geometry. So if you take Euclidean geometry and you measure, suppose a cell, they call it, you know, I measure 20,000 genes, all there is. It's in effect, I'm doing an embedding in a 20,000 dimensional Euclidean space. So the data will not be uniformly distributed there, but if I, you know, and then I embed this data, if you, okay, Euclidean data, you put it, you make number of measurements from 5D to 100D, and it's okay. And then you put it in the space, I think it's a 5-dimensional Euclidean space, you can recover the distances well. If you put this data after that into hyperbolic space, there will be a deviation. Now a case that turns out to resemble data better. If my data lives in a hyperbolic 5-dimensional space, if I put it in a hyperbolic Euclidean space, I will have a deviation initially, but after that, I flatten the data. After that, the data will appear Euclidean. But if I take the data, 5-dimensional hyperbolic space, put it in a 100-dimensional Euclidean space, I can preserve the distances through non-uniform distribution of points. And after that, if I put it in a Euclidean MDS, I will have a distortion. And if I put in a hyperbolic space, I will not have a distortion. So by focusing, as long as I measure 100 genes, I can use this as an indicator whether the space is hyperbolic or Euclidean. Any questions about this? Yes.
Is the 100 genes, is that like generally applicable, or is that, would that only work for the specific simulated data?
So the idea is, suppose that the, you know, foreshadowing the results, suppose the data belongs from a low-dimensional hyperbolic space, meaning the organism, you know, take a plant, for example. The plant can't do many different things at the same time. It can grow, it can bloom, it can defend against pathogens. Maybe 3 or 4 programs can, and they're mutually exclusive. So the decision-making is limited to 4 dimensions. And this is expressed in 20,000 genes. If I measure 4 key genes based on my biological knowledge, I will be able to capture the space well with 4 genes. But if I measure 4 random genes, I will flatten the space. So if I measure, so based on these simulations, if I measure 100 or more genes, I can have hope of reconstructing underlying 4-dimensional hyperbolic space. But if I measure less than 100 genes, so maybe 10, then my ability to recover underlying structure is diminished.
Does it depend on the depth of the tree? You get this tree structure.
Yes, it depends on the tree. Yes, it depends on the tree. So in this case, So this is data in 5 dimensions with a curvature, I think 5. Okay, but of course this depends. This is not a theorem. So in practice, for the that we think that 100 genes can be good. So this is not a strict result. It's just an intuition for how many genes you can expect to measure in order to, and then it turns out that, for example, if you denoise the data, what in our analysis, you denoise the data and the hyperbolic effects get slightly stronger. So noise will dampen it. I hope that's okay. That is what it is. So now this is analysis of gene expression, and this is synthetic points, and this is from real cells. So this is cells from all over different parts of the human body. And one can see that if you put the data into Euclidean space, you will have convexity positive. And when you put into hyperbolic space, the second derivative is close to 0. And it matches what you would get if you put synthetic hyperbolic points. Now, if you take data from a small, so this is the question of how deep is the tree. If you take data locally from a local sample, then the data is better described by Euclidean geometry than by the hyperbolic geometry. I hope that answers. So the conclusion was from this analysis of gene expression is that locally it is Euclidean, but globally it is more hyperbolic. And if you denoise the data, the curvature values increase by, say, 10%. All right. So this is how we, using this tuning procedure, how we determine the curvature of the space from human samples of about 2.5. And this is across different parts. So this is mouse embryo, has a relatively weak curvature, and differentiated cells have larger curvature. The brain has more curvature, and different types of human cells have a larger curvature. So you could use this hyperbolic depth as a measure of complexity in the data. Okay. Now, as I mentioned, there are different, you can put hyperbolic geometry in different ways. So this is the so-called method called Poincare map. So some of you know about t-SNE and how to use it to visualize data. So in this case, they use hyperbolic t-SNE with hyperbolic metric. We also use t-SNE. We also use hyperbolic metric. But the difference between the left side and the right side and is here that here hyperbolic geometry is global. In this case, global here, local here. So, and you can get a better preservation of distances if you do it in this way with its global, hyperbolic, local Euclidean, and embedding distances. Otherwise, you will have the bulge here. Any questions? We have 2 more data slides. Okay, so it's a little bit more on this hyperbolic t-SNE comparison with UMAP and some other method. But what I was encouraging here, so this is a data sets of different cells in the human body, and these are cancer cells. And what is interesting is that some cells are closer, have smaller radius, and they're closer to the center of the space. And you might know that cancer cell can also be of different kinds. And some of them are called cancer stem cells, the ones that can regenerate other cancer cells. And it's interesting that maybe you can use it as an indicator of being a cancer stem cell if you have a smaller hyperbolic radius. All right, so then one slide on COVID evolution, and then we will be done. So we looked at data on COVID. So then it's a viral evolution, so hierarchical process. So you can also verify that hyperbolic space fits better than a Euclidean. And what was very interesting here is that this is the curvature of the space as a function of sampling window, and the curvature increases as a logarithm. And then what was very interesting here, the last data point, the curvature actually decreased. And we were puzzled for a while until realized that this is where the vaccine was introduced. So the space grows, 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 and then you introduce the vaccine, and the space shrinks. So with this, I think I will conclude. I will show you pictures of hyperbolic geometry. This was a map of orderance that I skipped. This is human perception. This is this gene expression data that I discussed. We talked about neural representations and how they grow by adding more detailed representations, and how the radius increases with the logarithm of information. And we think it can, if one believes that hyperbolic geometry is relevant, then what we see is that it can be useful for data visualization. So this is hello from my group. The person who did original hyperbolic geometry in plants is Yuansheng Zhou, and he also did it in gene expression. And Honchou Zhang did hyperbolic geometry in neural responses, and Anup Raturu did COVID evolution. And this was collaboration with Brian Smith on orderance and Albert Lee on neural responses. Thank you for your attention.
Time for a few questions.
Since you have this relationship between learning and the hyperbolic geometry, lately a bunch of people trying to examine how do we learn, how to construct a curriculum to teach a person something. So do you think that curriculum basically exists also sort of geometry like that?
Yes, because I think these hierarchical models are the models for the language acquisition. So babies learn a big concept, and then, like a pet, and then there's a cat and a dog. And from my own personal experience, so when I was switching from condensed matter to neuroscience, I had all these papers on condensed matter, and I had one folder called neurons. And then that folder neurons grew, sensory and motor and things like that. So that's a canonical model for language acquisition. And people use it in machine learning mostly. So the work in machine learning is, I think they mostly use it because it gives better predictions with smaller number of variables. I'm more interested in it because it to create a map that you can assign parameters to this map that have biological meaning, such as it grows. But what I didn't talk about, for example, in the case of smells, you can have trajectory as the plant ripens, or if it is infected by fungus, the curve will shift. So you can have some trajectories there that are associated with biological processes.
This is I was wondering, how does geometry change if you aren't necessarily concerned with like how efficient the information passes, but if you may be that, how does this, I guess, change the?
Let me repeat the question such that I understand. So you said that, to my mind, the efficiency of transmission is related to robustness, but what we were interested in doing, but I did not fully investigate, but what happens with, say, traffic jams? Does the geometry flattens or not? So that I don't know. So I was worried that I think this is a well-studied literature or a large literature that I don't fully know. So there might be results there.
Yes, just one quick question. When they do the experiments and the mouse spends time doing something else, does the forgetting go, does the charts go backwards, or does like the decay of the information just have to do with how else the mind was used in between?
The forgetting is an interesting question. So what happens to the place that kind of atrophies? I don't know, but I think of this as a kind of a broccoli type growth. So we modeled it as a constant curvature, but in reality, the curvature is not constant. So it will grow in one part and have a higher curvature there. And maybe the neurons will flatten in another part and the neurons will be reallocated. That's a hypothesis.
You talked quite a bit about the hippocampus, where perhaps we expect to see some efficient tiling of some space that you want to represent. Do you expect, or well, empirically, well, I guess two questions. Empirically, do you see, or would you expect to see similar hyperbolic geometries in like other regions of the brain?
Yes, absolutely. I think wherever we look, we do find it. It's the question of different dimensionality and different curvature. And the question for mathematicians, I think, often we see a three-dimensional hyperbolic geometry. Although it's not a, it's not an absolute claim. It could be 3, 4, 5-dimensional, but two-dimensional geometry does not fit well. Starting with 3 and higher, it often fits well. In the case of COVID, it was more high dimension. More questions?
When growing neural net, is there any trade-off between increasing the size and changing the curvature? And what determines which way to expand?
I don't know. So it's often difficult to disentangle the two, the dimension versus a size. Our analysis on COVID data showed, so my bias is that I think that it will grow primarily in curvature and not change dimensionality. So this is like Bayesian information criterion as a function of dimension for different, as a function of time. So the effect on curvature is larger than the effect on dimension. So dimension, I would say, doesn't change much right here. So, this minimum is a function of dimension. So my interpretation of the data is that there is a weaker effect on dimension than there is on curvature.
Dimension meaning size, not the dimensionality.
No, dimensionality. Dimension, yes. So
Size.
And the size is dimension, and so let's talk about size. The size changes, but the dimensionality stays approximately constant. And I'm hoping to interpret dimensionality as the number of independent variables that describe the space. That's possible.
Okay, I have more questions, but maybe we should
Thank you.
you again.